Wet age-related macular degeneration is characterized by the presence of choroidal neovascularization. Evidence suggests that chronic risk factors can lead to the overexpression of VEGF and the formation of CNV. The direct link between the overexpression of VEGF and the pathophysiology of wet AMD is key to understanding the chronic risk of recurrence of CNV throughout the entire disease continuum. There are two main causes of the overexpression of VEGF in wet AMD: inflammation and hypoxia. There are several chronic risk factors that contribute to inflammation and hypoxia. Oxidation is one of the main causes of inflammation. In wet AMD, research has shown that oxidation occurs as a result of UV light exposure and aging. UV light exposure results in the presence of photooxidation end products, while aging leads to the accumulation of advanced glycation end products, or AGEs. The presence of AGEs and photooxidation end products can stimulate processes that trigger inflammation. The inflammation then activates the secretion of VEGF by the RPE. The complement system also plays an important role in inflammation and wet AMD. The complement system functions as part of the body's innate immune system to provide protection against infection. When triggered, a biochemical cascade is initiated that amplifies the immunological response and destroys bacterial and viral cells. Complement factor H, or CFH, is a critical inhibitor of the complement pathway, downregulating complement activation, potential inflammation, and tissue damage in response to immunological triggers in wet AMD. Normal CFH proteins bind with activated complement to prevent inflammation. Recent studies have demonstrated that a CFH gene variation can account for 50% of the risk of developing wet AMD. The CFH gene variation in wet AMD interferes with the ability of CFH to inhibit and bind with the activated complement on cells of the retina, the Bruch's membrane, and the choroid. Therefore, mutation in CFH fails to downregulate complement-mediated inflammation, leading to overexpression of VEGF and/or direct damage to RPE cells. In wet AMD, this overexpression of VEGF caused by uncontrolled complement within Bruch's membrane and the RPE leads to the formation of CNV. Like inflammation, hypoxia is a major cause of overexpression of VEGF in wet AMD. The choroid is the main source of oxygen and nutrients for the retina and is also the only source of blood supply to the fovea. Bruch's membrane naturally thickens over time, separating the RPE and outer retina from the oxygen diffused from the choroid. Research has also shown that the capillaries of the choroid may be smaller than normal and fewer in quantity in patients with wet AMD, which reduces blood supply. Hypoxia leads to overexpression of VEGF by the RPE, which can cause irreversible damage. In wet AMD, VEGF plays a significant role in the formation of blood vessels that grow abnormally and leak beneath the macula. Overexpression of VEGF initiates the abnormal growth of choroidal blood vessels through Bruch's membrane and into the subretinal space. These choroidal neovascular vessels leak blood and fluid, forming the characteristic lesions of wet AMD. Due to the chronic nature of these risk factors, patients with wet AMD may be at constant risk of recurrence of leakage, even after months of being dry. Therefore, consistent control of VEGF can be essential to minimizing the risk of recurrence of CNV in patients with wet AMD.